Hi, my name is Emmanuel Ogbodo. I'm a specialist biomedical scientist and I'm also a lecturer in biomedical science here in the United Kingdom. I completed my PhD in biomedical science. I have extensive experience working in an NHS hospital as a specialist biomedical scientist. I have used these experiences to help a number of people navigate through interview questions and therefore get their dream job as a biomedical scientist. I'm here to help you navigate interview questions, thereby increasing your chances of getting a job as a biomedical scientist. What I would ask is that you like, share, comment, and subscribe to our page. Thank you very much.
Hello guys, so I'm back again today because I really want to finish this antibody panel. So you can see the skill of identify antibody panel is increasing. Okay, but let me just encourage you. I don't want all of you to be overwhelmed. I can guarantee you that if you understand what I had said in part 1, part 2, and part 3, now in part 4, that is the hardest it can be. If you understand it, once you go for interview, it doesn't matter what they present, you will be fine. Because in the interview, it's not going to be as difficult as, I'm going extensively to explain these things for you guys. Because when you start working in the lab, you will know that this information, they are very vital. Okay? Anyway, so let's get into this chapter 4. This part 4 rather. So in this part 4, I'm going to mainly focus, I'm going to focus mainly on heterozygous antibody and homozygous antibody. So in this part, you are going to understand what we mean by homozygous antibody, heterozygous antibody. I know I had highlighted it a little bit in part 3. Now let's get into it deeply, okay? So again, we are going to do antigram body identification, whichever one, interpretation, that is what we are doing, okay? So I've explained this dosage, but if you've not watched my video, especially in part 3 now, to understand this dosage, you need to go and watch part 3. But I would suggest that please, if you've not watched part 1, part 2, part 3, please watch them before watching this very part 4, okay? Again, what I was trying to say in the dosage effect is that where antibody, you can see, is only JK here, only JKB here. At this point here, they are homozygous. JKB is homozygous antibody here. JKA is homozygous antibody here. JKA and JKB, they are present here, therefore it is heterozygous antibody. And I said that there will be competition. They may not bind strongly. The reaction may be weaker. And in being weak, it might even be negative. It does not mean that the antibody is not there because of that competition. Does that make sense? So that is what you've got here. So, and I'm saying when we are doing cancellation method, don't cancel where it is heterozygous. Because even if you get negative on the patient sample, for the fact it is heterozygous on that particular number of cells, don't cancel. You should only cancel at the homozygous where the antibody is positive on the homozygous, but the patient reaction is negative. Then you can cancel. I think I've made that point already. So let's go straight to this. I've just highlighted things here for you. So where it is highlighted green, they are homozygous antibodies. Where it is highlighted purple is what? Heterozygous. Let me give you an example. I'm going to leave you to study this, but I'll give you an example. In terms of heterozygous, don't worry about D. I want you to worry about big C, small c. So I highlighted on cell number 1. I'm not going to go through all of them, but I want you to study it. I've highlighted cell number 1, green. So I want you to look at this big C, okay? Here, when it comes to in cell number 1, in cell number 2, big C is positive and small c is negative, both in cell 1 and cell 2. Therefore, big C in cell 1 and 2 here is homozygous. Okay? Now, let me give you another example. If you come to E in cell number 3, E is positive, small e is negative. Therefore, big E is what? Homozygous antibody in cell number 3. In little c is positive in cell number 3. Look at big C is negative in cell number 3. Therefore, little c is homozygous antibody. And if you look at all these places, little c is positive, and big C is negative. That means all these places, little c is homozygous antibody. Then if you look at little e is positive on cell number 1 and 2, and big E is negative in cell 1 and cell 2. Therefore, little e is homozygous in cell number 1 and number 2. So it's homozygous, okay? Then and so on. Now, let us talk about where it is heterozygous, okay? Now, if you look at big C is positive in cell number 4, look at small c is also positive in cell number 4. So at this cell number 4, big C positive, small c positive, it is heterozygous antibody, okay? Now look at big E. Big E is positive on cell number 5, and small e is positive on cell number 5. It is heterozygous antibody. So where it is heterozygous is where I have told you, whatever thing you see in the patient result, even though I didn't put any patient result here, don't cancel if it is negative. Wherever it is heterozygous, don't cancel. Only cancel where it is homozygous, okay? So let's continue. Look at K. You will see that big K and small k, they are positive in cell number 2, so they are heterozygous here. But in cell number 1, small k is what? Homozygous. So you can see again in cell number 6, big K is what? Heterozygous with what? Small k, okay? So then and so on. Now let's go to Duffy A and Duffy B. You can see that in cell number 4 and 5, both of them are positive, okay? They are heterozygous. But look at cell number 1, Duffy A is positive, Duffy B is negative, therefore Duffy A is homozygous here. Look at cell number 2, Duffy B is positive, Duffy A is negative, Duffy B is homozygous. Then another family we are going to look at is JKA and JKB. You see that JKB is positive in cell number 1, JKA is negative, so that is what? Homozygous. JKB is homozygous here. JKA is homozygous in cell number 2 because it's positive, and JKB is negative. But look at cell number 3. Both of them are positive, and that is heterozygous. Again, you see it in cell number 6. JKA and JKB, they are heterozygous in cell number 6. I hope we're understanding this. Another family you need to look at is M and N, okay? You see that M and N is heterozygous here because they are all positive in cell 1 and 2 and 3. But look at M is homozygous in cell number 4 because it is positive and N is negative. And N is homozygous in cell number 5 because it is positive and M is what? Negative. Now another one, which is the final one, is big S and small s. You can see in cell 1 and 2, both of them are positive, therefore it is heterozygous antibody here. Then if you go to cell number 3, big S is positive, small s is negative, therefore it is homozygous antibody. Then if you come to cell number 4, okay, small s is positive and big S is negative, therefore it is homozygous antibody. So with what I've taught you, you can use it to study and see why I highlighted where I highlighted green or pink or purple, as the case may be. So you will see it is either homozygous if it is green, or heterozygous if it is pink or purple, okay? So here you go. That's the background I want to give you guys. So now, this antibody that I want to identify here, I've highlighted the result for you. So this patient, so I'm not, because if you've not watched part 3, watch part 3, because everything I'm saying here, I'm saying it with the mindset that you've watched part 3, okay? So look at this now. This patient has got antibody big E and antibody Duffy A, and we cannot exclude M, we cannot exclude big S. Now, what I want you to look at, you can see that big E is positive here, especially if you look at the enzyme matches big E. So that definitely confirms that there is big E. But look at Duffy A, wherever it is positive, okay? Even so, it is negative in cell number 2, it is negative. Even though it was negative in cell number 3 and is positive in cell number 3. Remember, you cannot cancel that way, but if you go to E, E is also positive, okay? That shows why it is both anti-Duffy A and anti-E, okay? So anyway, if you use that method down, you will see you cannot cancel Duffy A. But if you look at M, the problem with M here is that we could have cancelled M in cell number 2, but we cannot, from what I've told you, because of what? It is what? Heterozygous here. If you come to big S, we could have cancelled big S in cell number 2, but we cannot because it is what? Heterozygous here. Does that make sense? Okay? Now, but look at M. it is homozygous in cell number 4, but this is showing weak reaction. I highlighted it this way because even with this, it can give me a strong indication that this is not M, because if it is M, it's supposed to be a strong reaction here, not a weak reaction. But let's not worry about that. So at this point, if you follow, so where I highlighted red is where I excluded it. For example, in D, it's positive in cell number 2, and this is negative here, I exclude it. Big C, positive in cell number 2, this is negative, I exclude it. So what I want you to see, watch where I highlighted red, that is where I excluded the antibodies, and the antibody that is red, painted red up here, are the ones that I've excluded, okay? The one that is green is the one I believe is the antibody. So the one that is the light green, they are the ones I cannot exclude at this point. So at this point, we are very sure that it is anti-E, but we cannot exclude Dufi A, M, and S, but with a strong indication that it will be what? Dufi A. Does that make sense? But again, I want you to study this. Remember, in cancellation, you can cancel either on the IAT or you can cancel with the enzyme. But when you are cancelling with the enzyme, please don't use Dufi A and Dufi B or MNS on Dufi A on enzyme, because they are destroyed by enzyme. So when you are cancelling MNS and Dufi A and Dufi B, use only IAT, the one that is not enzyme treated. Okay, so now at this point, we are sure that from the enzyme it is E, but we cannot exclude Dufi A, anti-M, and big S. Remember that if we use the initial cancellation method I told you, we could have excluded we could have excluded M and big S. But because of now I've told you about homozygous and heterozygous antibody, unfortunately, where we're supposed to exclude M and big S, they were heterozygous, so we cannot exclude them there. So what are we going to do? You do phenotype. So you do phenotype, but I'm going to explain that more when we get to that point. So let's look at case B. So case B here, you are going to see that in this case B, you will see that I'm saying is anti-K and antibody Dufi B. Again, use the same method I've shown you. So from the enzyme, obviously it is K. Look at it, K. But again, the problem with K is that it is heterozygous here with small k. But don't worry about it. Then, if you look at, so the enzyme matches K exactly. It doesn't match small k. So it is antibody K. Now, look at the rest. Dufi B, you are going to find out that Dufi B is negative, positive, 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 positive. But look at Dufi B. Dufi B is positive here, but this is negative, positive here, but this is negative. So now let's look at that. So now, what I was going to say was that if you look at this, even though that Dufi A, Dufi B is positive, but it's negative here. But I've told you guys, because it is heterozygous antibody, that is why you are seeing this negative. That's why I say, once it is heterozygous, the competition will be high. And that competition can give you a negative result. Okay? So don't cancel where it is negative, but the antibody is heterozygous. So we cannot cancel Dufi B here. Okay? But if you look at Dufi A, we cancel Dufi A on cell number 1 because it's what? Homozygous, and it is negative here. Then apart from these places that Dufi B is heterozygous with Dufi A, the rest is positive wherever it is positive in the patient sample. Okay? Now, let's look at the place. So if you look at the places that I've highlighted red and what I highlighted red, they are the places that I cancelled this antibody. And what I want you to study from what I've told you previously, wherever that it is red is where it is what? Homozygous antibody positive, and the result is negative. For example, JKA is positive on cell number 4, homozygous, and this is negative, and that's why I cancelled it. Why do I cancel JKB on cell number 1? Cell number 1, it is positive, that's homozygous here, because JK is negative and this is negative, and that's why I cancel. But look at M. M is what? Heterozygous with N here, so I can't cancel it there. So, but I cancelled M on cell number 4 because it is homozygous on cell number 4 because it's positive here and negative, and N is negative. So positive in cell number 4 and result is negative. That's why I cancelled M here. So why do I cancel N on cell number 5? It's positive and it is homozygous, and this is negative. So study for what I've shown you. You will then see why I say that this is antibody K and antibody Dufi B. Antibody K because the enzyme matches the K, antibody Dufi B because I cannot exclude antibody Dufi B. I can exclude Dufi A. Every other thing here has been excluded apart from Dufi B and K. So that is why it is antibody K and antibody Dufi B. Okay? Remember that why I could not exclude Dufi B here, even though this is negative, is because it is heterozygous antibody. When you go for your interview, don't let this kind of thing to confuse you. That you see negative, please don't cancel it where it is heterozygous. Concentrate on canceling where it is homozygous. Then the answer will appear. So that's what I've done. So you study where I've cancelled with red. So where I've marked heterozygous with green or purple. Like if you look at the place like big C, I marked purple here because in cell number 4, it is positive and little c is also positive. That's why I marked them, okay? So you will see again, they are heterozygous. So the place that is purple or the place that are marked green stretched like this shows you heterozygous, okay? While the other ones that are marked red, where I cancel it, shows you homozygous antibody. So if you use that methodology, you will see why this is antibody K and the B, okay? So now, look at the same example here, but in a different way. So I've made this one to be a little bit more complicated, okay? So I make this to be more complicated for a reason, okay? So because of I had already taught you guys this, I'm not going to repeat myself over and over again now. So anywhere that I mark red, I've cancelled the antibody. So you study from what I've shown you, okay? Now, anywhere that I mark purple, like the way I've stretched this purple, I'm saying that they are heterozygous here, okay? Maybe with green, I'm saying they are heterozygous here. So now, if you look at that, you will see that we cannot exclude big C. Why? Because anywhere that it is positive in big C, it is positive here, okay? And even in the enzyme. And this antibody, you can see there are more reaction here. There's also more reaction here. So it's not like, it seems like what is here is not the antibody that is destroyed by enzyme. That's the point. That's why I try to put this. So unlike what I've been giving you previously, this one is not the antibody. There are 2 antibodies here, and 2 of them are not destroyed by enzyme. That's what I've put you here. So if you look at this, we cannot exclude big C. We cannot exclude CW. We cannot exclude BK, okay? We cannot exclude JK, but the rest can be excluded. All these antibodies that cannot be excluded cannot be destroyed by enzyme. You see it now. So now, because we now say, oh, it could be big C, it could be antibody CW, it could be antibody K, it could be antibody JK. What do we do? What we do then, like I was trying to say in the previous slide, is to do phenotype. Remember what I taught you guys. When these antibodies, they are measuring. Remember that any IgG antigen that can be found in the surface of the red cell of any patient, the patient should not develop the antibody. If the patient develops the antibody, that is autoantibody. So you are doing phenotype because if that phenotype is negative, it means the patient does not have that antigen. Therefore, the patient is likely to develop the antibody. But if the phenotype is positive, it means the patient has the antigen. You should not expect the patient to develop the antibody. If the patient develops the antibody, that is then autoantibody. So by this method I've shown you guys in terms of heterozygous, don't cancel where it is heterozygous, you will see that we cannot be able to cancel big C because anywhere big C is positive, the reaction is positive, okay? Whether in the IAT or in the enzyme, okay? And anywhere CW is positive, reaction is positive, whether it is in IAT or enzyme. And anywhere BK is positive, the reaction is also positive, whether in the IAT or in the enzyme. And anywhere JK, is positive, the reaction is positive, whether in the IAT or in the enzyme. So even if you are tempted to cancel here, remember what I say, please don't cancel because it is heterozygous. I know why I'm emphasizing this. I know this is 0, but I cannot cancel it because it is heterozygous here, okay? Anyway, so bearing that in mind, what we're going to do then is to do the phenotype. And here you go. This is the phenotype. So from this phenotype, we can see, remember that if you go back, these were the antibodies we are suspecting. We are suspecting big C, we are suspecting antibody CW, we are suspecting big K, we are suspecting JK. So now let's look at this. So big C is negative. We say because it's negative, the patient is likely to develop the antibody. Does that make sense? Therefore, that shows that it can be big C. Okay. Now CW is positive. Therefore, the patient shouldn't develop the antibody. That means it's not CW. And you can see that JK is negative, and because it's JK negative, that means the patient can develop antibody JK. Do you understand? And you can see antibody K is positive, and therefore, because it's positive, the patient is not expected to develop antibody BK. Therefore, you cannot exclude CW and antibody BK using phenotype, because their phenotype is positive and the patient should not develop the antibody. But big C and JK is negative. Therefore, that confirms that is the antibody, because the patient can likely develop that antibody. So when you go, you see the way I'm taking it now. So when it becomes difficult to exclude, you have tried the matching method, you have tried the cancellation method, homozygous or heterozygous, and there's still antibody that you are not able to exclude, you do phenotype. Phenotype will then help you to confirm whether it's going to be that antibody or not. So that is what I've done here. Okay. So let's look at this case D. In this case D, you are going to see that. In this case D, you are going to see that obviously this enzyme, because I'd already given you the information, so I'm going to be quick. This enzyme obviously match anti-D. So we know that there is anti-D here. Okay. But look at big C. We are not able really to exclude big C. By the way, wherever that is red, highlighted in red, they are the ones I'd already excluded. So we are not able to exclude big C because it's positive here, positive here, and of course positive here. Now, in cell number 4, it is positive, but in the I80, it is negative. But you cannot exclude it there because it is heterozygous. Look at it. Cell number 4, big C and small c, they are heterozygous, so you cannot exclude it. Okay, so that's why we are still suspecting big C. Look at big E, the same thing. Okay, with big E, you can see it's positive in cell number 3, positive here, and also positive here. And then look at when it comes to cell number 5, it's positive, but cell number 5 here is negative, but we cannot exclude it again because it is what? Heterozygous. Look at it, in cell number 5, positive, with little e, positive. So that's why we cannot exclude it. Okay, but having said that, we are able to exclude both little c and little e. How? Because in little c, in cell number 5 is positive, and big C is negative. That means little c is homozygous here, and this is negative. You exclude little c. Little e is positive in cell number 4. Okay, big E is negative. Therefore, in cell number 4, little e is homozygous. Okay, then you will then exclude it. Okay, because it's negative. Okay, let me repeat this again because I think I may have muddled it up together. So if you come to cell number 5, little c is positive and big C is negative. That means little c is homozygous. Then, and you look at the patient result, is negative. That's why you exclude little c. So you've seen how I exclude little c and little e, but I could not exclude big C and big E because where I was supposed to, they were heterozygous with the either little c or little e respectively. Okay, now if you come to CW, we cannot exclude it because where it is positive here and here. And K, we cannot exclude. Even if we are tempted to exclude it on cell number 6, we can't because of why it is heterozygous in cell number 6, even if this is negative. Okay, but we've excluded little k. Okay, then we've excluded Dufi A, Dufi B, and all the rest. So you can work it out from what I've said. So where excluded is where it is homozygous and it is positive and the patient sample is negative. So let me also give you another example like M. I could not exclude M in cell number 6, M and N, because of why they are heterozygous here, even though this is negative. The same thing with S. I couldn't exclude it in cell number 6 because it is heterozygous here with small s. Okay, but then if you look at M, I excluded M in cell number 4 because it is homozygous in cell number 4, and this is negative. And N, I excluded it because it is homozygous in cell number 5, and this is negative. Okay, while in little s, I excluded it in cell number 4 because it is homozygous. You can see it's positive, while big S is negative. So it is homozygous, and this is negative. So that's how I've been able to exclude the ones I excluded and the ones I didn't exclude because of heterozygous, as the case may be. But obviously here we know that this enzyme contains anti-D, but we are not excluding all this other. We are not excluding big C, big E, CW, and K, and S. That is why we then need to do phenotype. So look at what we are suspecting so far, but we will definitely know that there is anti-D. So we're going to investigate the rest, and we're going to do that looking at the phenotype. Okay, so that's what we're going to do. We've done the phenotype. So when you've done the phenotype, because initially we were suspecting all this. Okay, we already know it's D anyway from the enzyme. Okay, but we are suspecting the rest of this. So now you've done the phenotype, and following the phenotype, you can see that big C is positive. Therefore, it shouldn't be big C because it has the antigen, and big E is positive, so it shouldn't be big E because it has the antigen. CW positive, it shouldn't be CW, because it has the antigen. K positive, it shouldn't be K because it has the what? Antigen. Okay, so now if you look at that, you can now confirm yes that this is antibody D and antibody S, because of course S, the phenotype is negative. So that's what happens. So sometimes when there's a lot of antibodies you are not able to exclude, the next thing might be to do the phenotype. That way you can be able to exclude them. But during your interview, it shouldn't be this complicated. But I want you to study this because it will help you, especially when you get the job and start working. And in some cases during your interview, they may want to know whether you have the idea about this phenotype. Okay, maybe you said it could be this, it could be this, it could be that. They may just say, okay, they may not give you more panel to do. They may say, okay, what else can you do? Or they may even ask you, when is it required to do phenotype? I'm going to make a special video for that. But this is one of the scenarios when you are allowed, when you are supposed to do phenotype, because that will help you to actually know the antibody that the patient has got. So here you go. This patient has got anti-D and antibody big S. Okay? Yeah? So here you go. That is your heterozygous and homozygous antibody. So I'm not sure if you have any question. If you do have any question, again, I would like you to put your comment in the comment section, or you can message me, privately, and I'm more than happy to, of course, discuss further with you. And again, what I've done now, I've given you all the skills that you need to identify antibodies, whether it is one antibody or two antibody, whether it includes antibody that can be destroyed by enzyme or not. It doesn't matter whether necessary where it has to do with doing phenotype. I have also included where it is pan-reactive, when you need to send it to the Arusia, or you call it non-specific antibody, whichever way. So I've explained all this whole thing. What I will advise them is that you go and listen over and over part 1, part 2, part 3, and this part 4. And then test yourself with any antibody panel anybody can give to you. And I tell you, you have now become an expert if you understand everything I've said. Thank you very much till I come back your way again. I would also ask, please, can you hit the subscribe button and share to your friends and family? Thank you. Bye.